Hello, this is Dr. Lucy Abgarian and this is class Bio 225 Genetics from Genes to Genomes. Today we are going to cover Chapter 7, Part 4. Now let's talk about experiments, tests that were suggested and conducted in order to find mutagens. Particularly the AMES test is introduced to identify some potential carcinogens, and actually it was used to determine many carcinogens. So what's the experiment? The idea is that one takes S. Typhimurium bacteria, which is prone to mutations. It has a great potential to be mutated. One significance about this bacteria is that it is growing only in histidine-containing medium. So carcinogens are introduced to this bacteria, and if mutations take place, it will affect the ability of this bacteria to grow also without histidine. So if there is a mutation, even from so many genes, only few are mutated, they will continue growing if we have a media where bacteria is existing without histidine in it. And as you can see, only if there are only 2 genes that contain, they will grow a colony. And so that's how the carcinogens will be identified as mutagens. So what can mutations tell us about gene structure? The complementation testing. So let's say we have organism that has mutation on a recessive gene. So if we are dealing with homozygote, then we will see mutation for sure. If we are dealing with heterozygotes, then we will not see the mutation because we have also the I have to mention here that the mutated gene is the recessive gene. To take that's very important point. So in case of homozygotes, we will see the mutation. In case of heterozygotes, we will not see the mutation because the mutation is on recessive gene and the dominant gene is not carrier of the mutation. Therefore, in heterozygotes, we will not see the mutation. What is the complementation group? Let's say if the mutations are on different genes, for example, enzyme A and enzyme B, they cause certain eye color. So if there is a mutation on A, we will not see the eye color. If there is mutation only on B, we will not see the certain eye color. If mutation is on both A and B, we will not see the eye color. The only case where we will see the certain eye color is when there is no mutation neither on A nor on enzyme B. So that's how the complementation group should be associated. So let's talk about our favorite Drosophila eye color mutations. So in case of complementation, if the gene defect, the mutation is in 2 different genes, and we have for both of the genes we have a functional copy, then we will see in offspring we will see the wild type gene. in case of no complementation, where the defect of the gene appears in both copies, the M1 and M2 defects both are on gene G, then we will not have a functional G gene in offspring. So now let's talk about it on an example. So here, the first picture is the eye color of the Drosophila with the wild type. The second one is without any eye color, which is the case of no complementation, where the mutation appears on both copies of the same gene, then there is no eye color. And the second and third cases are the cases where the mutations are in two different genes, the complementation case. So still there is an eye color. Of course, it's not the original eye color. And I mean, and it's because the Drosophila eye color is it's a complex transfer of the genes, causation of the genes. Okay, now let's look about and see the Drosophila eye color mutations and how they are affecting due to complementation. So the eye color is X-linked gene, and the mutations can result in like either 5 complementation groups, or they can be not affecting others at all. So let's look at that. So for example, mutation in white, cherry, coral, apricot, and buff, as you can see on the table, are mutually connected. So mutation in white will cause mutation defect in cherry, coral, apricot, and buff as well. The same is with cherry. It's correlating with white, coral, apricot, and buff. The same with coral. It correlates with all above mentioned, and the same is with buff. So that's when the complementation groups genes take place. However, there is also mutations that are independent and they don't affect anything else, such as carnation, for example. It will not affect any other color. So those are independent enzymes that are being mutated that cause this particular color, such as garnet, ruby, vermilion, and carnation. So fine structure mapping of phage T4 mutants. Benson's work was fundamental to determine whether can recombination take place between different mutations in the same gene, intragenic recombination. And if you remember from the previous classes that we have talked about recombination, the closer the genes were, the fewer were the occurrence of the recombination. So in particular case, Benzer did the experiment with phage T4 virus, which is a virus that is affecting E. coli. And the reason that this virus was picked is for mass structure mapping is because it's producing progeny, like hundreds of progenies, from 100 to 1000 in less than an hour. So there will be a large number of progenies to detect rare events, like mutated progenies. And conditions, these conditions will allow proliferation of only recombinant phages and death of parental phages. So this was very convenient tool to use in order to answer this question. Now let's look actually what does that look like. So looking at the original chromosomes, we can see that the two genes have mutations at two different places, mutation M1 for one of them and mutation 2 for another of them. When recombination takes place, um, we all of a sudden end up having a recombinant gene 1 which contains both mutations M1 and M2, and another recombinant gene which is ending up being the wild type, not carrying any of the mutations. Ta-da! Magic happens, and we end up having gene wild type with wild type sequence in it. This considers one of the fundamentals of the subject, and it's worth to look at the bacteriophage T4. Here on top left is presented the electron microscope image of bacteriophage T4. Next to it is the cartoon representation that shows the entire thing with the viral chromosome in it, and then with the sheath, and then also the tail fibers on the bottom. There is the representation of the cell cycle, phage injected, of how the phage injects in DNA. Hosts like its DNA into the host cells, how that is happening, and then a second step of the cycle where the phage proteins are being synthesized, DNA replication takes place, host chromosomes degrade, and then the assembly of the phage within the host cell, and then finally the lysis of host cell. And then on the bottom left, you can see how the petri dish with the infected bacteriophage T4 looks like. And here is a representation how the counting of bacteriophages done. It's done by serial dilution. So we have the concentrated solution with the bacteriophage in it. We do serial dilution, pipetting out 10 microliter of the original to the 1 milliliter, and then 10 microliter of the second dilution to the third one, which contains 1 milliliter, and so forth, until we have diluted sample, and then we add that plating bacteria into a petri dish, and we get cultures, colonies, and based on that amount of colonies, we can back calculate our original stock and the concentration of our original media containing the bacteriophage. So what was the Benzer's experimental approach to find structure mapping of the R2 locus? So he generated 1,612 spontaneous point mutations and also several deletions in R2 locus. And in order to do that, he identified two complementation groups, R2A and R2B. He mapped locations of deletions relative to each other using recombination. Also mapped locations of point mutations relative to the deletions. And also tested for recombination between all point mutations within the same complementation groups. In case I have confused you at the previous slide, let's clarify what was I was trying to say. Okay, so let's look at this in this cartoon representation. So first case, we are talking about complementation test in trans configuration, which means that we have two mutations of R2 two different mutations. So by infecting E. coli, we will end up having situations where we can have either non-functional R2A and functional R2B, or we can have functional R2A and functional R2B, which means that in any case, we are getting 100% R2B and we are getting 50% R2A. So, which means in case of no complementation, we will not see cell lysis and also phage progeny. In case of complementation, we will see both the cell lysis and phage progeny. So let's look at the control, which is in cis configuration, which means that our mutation, both mutation 1 and 2 are occurring on one gene, and another gene is remaining wild type without any mutations. So which will result in two scenarios. In case if the mutations were on recessive gene, we will end up having cell lysis, which means it will not affect our progeny. We will end up having wild type healthy gene. In case of the mutations being on a dominant gene, we will not have a cell lysis, which means the mutation will affect the process and the gene. So this was all the complicated talk explaining the complicating conversation we were having before. So now we can, when we clarified things already a little bit, now we can also clarify the part with the recombination. So when we say recombination test, we mean then we have R2A1 and R2A2, both with mutations. And due to recombination, we can end up having R2 wild type, or we will have R2A1 plus R2A2, which will contain both mutations within. And subsequently, it will result in having formation of plaques on E. coli, or no plaques in case of both mutations. So it will give us a chance of having healthy wild type gene without any mutations on it. While when we are looking on the control, when we have the one mutation or the other mutation, then in both cases there is no chance for no plaque without recombination. Today we have talked about mapping for point mutations. Next time we will talk about mapping while using the deletion.